Today's topic is the cytoskeleton, which I assume you've probably learned something about before, but I'm going to focus today on microtubules, but as an example of cytoskeletal filaments that are part of the network of different filaments that make up the cytoskeleton of the cell. So when we talk about the cytoskeleton, we basically mean these filamentous structures that have varying degrees of elasticity and rigidity and resilience that give cells their structure, their ability to move, their ability to change the location of internal components. And there are three major classes of cytoskeletal filaments that we'll talk about, and I will focus, as I said, on microtubules. These cytoskeletal filaments are polymers. That means that they are made up of individual proteins that get put together into some kind of multimer or assembly that gives them their properties. And there are different proteins that make up intermediate filaments, microtubules, and actin filaments, and they play different roles in the cell. We tend to focus, as cell biologists, I think there's more attention paid to microtubules and to actin because these are both highly dynamic networks, so they're constantly being built and broken down and changing shape within the cell. By contrast, intermediate filaments are a little bit less dynamic. They have to be built and reassembled whenever a cell divides, for example, but they don't tend to be quite as dynamic. Also, as I'll talk about, actin and tubulin, the proteins that make up actin filaments and microtubules, are enzymes, which imbues them with dynamic properties that are lacking in intermediate filaments. So I'm going to just mention a couple of points about intermediate filaments and then move on to microtubules. This shows micrographs of typical epithelial cells with the different filamentous structures highlighted in different colors. And the book is pretty good about sticking to green for microtubules and red for actin, and I've tried to do that in this as well. The key thing here to point out is that all of these are made of proteins. The proteins, so intermediate filaments are more diverse. There's quite a few different kinds of intermediate filament proteins. We've talked about one, which is lamin, that makes up the nuclear lamina, but there are also cytoplasmic filaments and filaments that connect cells. I'm going to focus on microtubules in part because we're going to talk about mitosis and meiosis later this week, and microtubules play an essential role in cell division and specifically in pulling chromosomes apart during mitosis and meiosis. And then at the end, if there's time, I'll talk a little bit about actin filaments. So intermediate filaments are called intermediate because they're intermediate in width between sort of fat boys, the microtubules, and the thinner actin filaments. Actin filaments, even though they're thinner, they still play a major role in giving cells structure and resilience. The only thing I was going to point out about intermediate filaments is they have a different kind of structure than microtubules or actin. Both microtubules and actin are made of globular subunits that stack together to make a polymer, whereas intermediate filaments are made of these long proteins that have long stretches of alpha helical structure. And one thing we've talked about alpha helices in the context of membrane proteins, that alpha helices form these rods that can go through a membrane. But alpha helices are a very flexible kind of protein structure. And one thing that they can do is they can wrap around each other to form what's called coiled coils, because it's a coil of coils. And intermediate filament proteins have this kind of structure, and they can wrap around each other like this, where their N termini are at the same end and C termini are at the same end. That's called parallel. Coiled coils can also form in an anti-parallel fashion where the N and C termini are at opposite ends. And then these coiled coil structures can then interact typically through domains at the heads and tails of the coiled structure to build up bigger networks. And so that's how, for example, the nuclear lamina is constructed. Lamins have this kind of structure, this coiled coil structure, and then they interact with each other to form this cross-linked network. This is one thing that the book points out, is that there are different kinds of cytoplasmic intermediate filaments in different kinds of cells. They're specialized for different kinds of cell functions. All animal cells have nuclear lamins, but they actually vary a little bit depending on the cell type and what's going on in those cells as well. And I'll talk about that a little bit more when we talk about meiosis. Just to remind you that I've already introduced the nuclear lamina. This, again, is a scanning electron micrograph of a preparation of a nucleus that's been open-faced. And then this is coated with metal and imaged by scanning electron microscopy. And it shows this network that's just shown as these kind of scribbly filaments at the nuclear envelope here. And as we talked about, they interact with chromosomes and they give the nucleus shape and rigidity, and mutations in the structure lead to a wide variety of diseases. And I've touched on that in previous lectures. Now I'm going to turn to microtubules, which I think are the most exciting filaments. If you were taking this course from David Drubin, it would probably lean more toward actin. It's just a question of what we think is interesting and what processes we care about most personally inside the cell. But microtubules play a number of key roles in cells. They are organized by something called microtubule organizing centers, which I introduced in a previous lecture. It's not so obvious in this image, but the microtubules in this non-dividing cell, in this interface cell, are mostly concentrated here at the site adjacent to the nucleus, and they're radiating out toward the cell surface from a microtubule organizing center that's not stained here, but it's embedded in the densest part of this microtubule network. So the microtubule organizing center has its own replication cycle. It varies in its organization depending, again, on what kind of cell we're talking about and whether the cell is actively dividing or not. In a non-dividing cell, the microtubule organizing center is typically adjacent to the nucleus. It's not shown here, but it's actually tethered to the nucleus. So this is a little bit inaccurate because it's actually stuck to the nuclear envelope in most cells. And then during mitosis, as I'll talk about on Wednesday, the microtubule organizing center has replicated, just like chromosomes replicate in S phase. The centrioles at the core of the microtubule organizing center replicate, and they split apart and form this structure that we call a spindle, a bipolar spindle, with the two poles defined by the two microtubule organizing centers. And then in cells that have cilia, which can perform a variety of different sensory functions within cells, the cilia have microtubules that extend throughout them. And they, again, have a microtubule organizing center at the base. In this case, it's called a basal body. In this case, it's called spindle poles. In this case, it's usually just called a centrosome or microtubule organizing center. Microtubules, as all filaments, are formed from a core protein, and in this case, two proteins that are very, very similar to each other and arose through duplication and divergence of the two subunits. So basically the same protein, but one's called alpha tubulin and one's called beta tubulin. Their ancestor was a GTPase.
Beta tubulin is still an active GTPase. It binds and hydrolyzes GTP to GDP. Alpha tubulin, on the other hand, has lost its GTPase activity. So it binds GTP but cannot hydrolyze it. So it just remains bound to GTP, and that's why the GTP is called non-exchangeable.
You might wonder why a filament would be built out of a protein that is an enzyme. And that's because this is a way that the cell can utilize the energy of GTP hydrolysis to regulate the assembly and disassembly of tubulin. Filaments are only useful if the cell can build them where they need them and break them down when they don't need them. The hydrolysis of GTP drives the assembly and disassembly of microtubules, as I'll talk about over the next few slides. Microtubules are hollow tubes, and this dimer of alpha and beta tubulin forms long polymers that are called protofilaments, but they're not stable by themselves. They co-assemble with other protofilaments to form this tubular structure, which is not completely radially symmetric. In most parts of this structure, the alpha subunits are next to other alpha subunits and the beta subunits are next to other beta subunits, but there's this line where they come together called the seam, where alpha and beta are juxtaposed. And so there's this asymmetry, and the cell takes advantage of that in certain ways to direct movement and to control the assembly and disassembly of this structure. A lot of what we know about the organization of the tubulin proteins within these microtubules came from work by Eva Nogales' lab at Berkeley. Her lab developed techniques to prepare microtubules and image them by cryo-electron microscopy. And that's what's shown here is microtubule filaments. These are unstained. They're imaged by this technique called cryo-EM. And using averaging techniques, the Nogales lab was able to go in and actually figure out how the tubulin protein is organized within these tubules. These are very rigid structures. They have the rigidity of plexiglass. A single microtubule, although it's only 25 nanometers wide, can grow extremely long, and they tend to be quite straight. You can see them bending a little bit here. But overall, they have a very rigid and straight kind of configuration within the cell compared to other filaments like actin, which are much more bendy. This shows
sorry,
yeah.
sorry, we have a question.
Go ahead.
So the slide, polymerization does not require GTP. Someone wants to know whether or not it is required for assembly and does that mean it's not required for assembly and disassembly, but it usually is coupled?
So that's a really good question that I didn't explain when I showed the slide earlier. Alpha and beta tubulin both bind to GTP, and in that form, this dimer can assemble into protofilaments, which then can form microtubules. And the microtubules are stable when beta tubulin is bound to GTP. When it hydrolyzes GTP to GDP, that destabilizes the dimer within the filament and leads eventually to the disassembly of microtubules, as I'll show you in a few slides. One way that researchers learned this is by purifying alpha and beta tubulin and showing that it could be assembled in vitro into microtubules in the presence of chemicals like GMP-CPP, which looks very much chemically like GTP but cannot be hydrolyzed because the gamma phosphate that would need to be hydrolyzed has a bond that can't be cleaved by beta tubulin. In those conditions, alpha and beta tubulin can still assemble, but they don't disassemble.
Both actin and microtubules grow into polarized filaments, so they have what's called a minus end and a plus end. And the plus end is where they tend to assemble more subunits. In the case of microtubules, the minus end is actually stabilized by a nucleating complex that I'll show you in a second. But the plus end is where most of the dynamics are. The microtubule organizing center nucleates microtubules. So what does that mean? In this case, it provides a template in the form of this protein complex called gamma-TURC, gamma-tubulin ring complex, which I'll show you in a second. That is the nucleating structure that a microtubule grows from. So what a microtubule organizing center is a high concentration of this gamma-TURC complex. Gamma-tubulin ring complex, as you might imagine from the name, actually has another form of tubulin, gamma-tubulin, so not alpha or beta, that form this split washer structure that is what templates the growth of microtubules. And that's true for all of these different microtubule organizing centers. So at the core of the microtubule organizing center is this structure called the centriole, which is actually made also of microtubules. And then surrounding the centriole is this cloud of material called the pericentriolar material. And that's where microtubules nucleate within the cell. They sometimes have other names. As I mentioned, in a non-dividing cell, the microtubule organizing center is sometimes called the centrosome. In a mitotic cell, the two microtubule organizing centers are sometimes called spindle poles because they form the two poles of the spindle. And when you're talking about cilia or flagella, the structure that nucleates the microtubules is often called a basal body. This shows a little bit more about the organization of microtubule organizing centers. At the core is usually this pair of centrioles, this weird structure made of microtubules that has its own replication cycle. And these centrioles then recruit proteins that nucleate this cloud of material around them called the centrosome matrix, or more often actually the pericentriolar material, or PCM. And this material has sort of a cloud-like appearance in the cell, and within it are a high density of these little ring complexes that are the gamma-tubulin ring complexes. And those nucleate microtubules, and the microtubules grow outward, adding to their plus ends, often until they get to the cell cortex, or if we're talking about mitosis, until they reach a kinetochore, and then they stop growing and are stabilized. And this is actually a micrograph showing the two centrioles at the core, these gamma-tubulin ring complexes in red, in the sort of cloudy material around them, and the microtubules emanating out from that. So this shows a little bit more clarity. This is an actual micrograph showing these two centrioles, and they're arranged at right angles like this. And again, they're made of microtubules. What controls their length and their bundling, I think, is not well understood. But they go through a replication cycle. So they have a mother and a daughter. The daughter was the newborn one, and the mother was the older one. And they recruit this material around them, including the gamma tubulin ring complexes. One thing that I just mentioned, because it's not mentioned in the chapter, but actually a former Berkeley graduate student went on in his postdoctoral work to show that the pericentriolar material is probably a membraneless organelle, that it is a condensate of some kind. The proteins within it are highly dynamic and they're moving around all the time. But part of what's embedded in this material is this crucial gamma tubulin ring complex. This is a cryo-EM of gamma tubulin ring complexes. So they're formed of gamma tubulin and other proteins, which together form this open washer structure, but it has the same diameter as a microtubule, and it's thought to form a template for the growth of these protofilaments and their formation into a tube to make a new microtubule. Sorry, what does nucleate mean? So yeah, I haven't really defined that, and it's a good question. So when you're talking about the assembly of any kind of polymer, in principle, you could have equilibrium between a polymer, meaning an assembly of many subunits, and a pool of soluble subunits. But in practice, the cell controls where the polymer assembles by nucleating, meaning sort of kicking off the polymerization process. And it can do that in different ways. So in the case of microtubules, it does that by providing this template structure that tends to overcome the energy required for these initial subunits to come together to start the polymerization to form a microtubule. There's usually some kind of energetic barrier to spontaneous assembly of filaments because the cell doesn't want them just forming in random places. And in the case of microtubules, the catalyst for assembly is the presence of these gamma tubulin ring complexes. In the case of actin, which I'm not really going to talk about in any detail, there are nucleating factors, often ARP23 or formins, that are activated at certain places in the cell, and that kicks off the polymerization of actin. I don't know if that explains the term nucleate, but I would suggest that you Google it and then come back to me if you want more details on the explanation. So I've alluded to this already, that microtubules undergo major changes in organization between different stages of the cell cycle. So this shows a typical interphase cell with the microtubule organizing center here next to the nucleus, and the microtubules radiating outward toward the cell cortex. And then in dividing cells, most of the microtubules are organized to form this spindle, although there are still these what are called astral microtubules that kind of come out the back end of the spindle pole and interact with the cell cortex, and they help to position the spindle in the middle of the dividing cell. This just shows an image of microtubules actively growing and shrinking. And you might notice, if you look closely, as researchers did in like the 1980s, that in the same cell, you can simultaneously have microtubules that are growing and microtubules that are shrinking. And that was kind of conceptually weird, because if you think about chemistry, you might imagine that if the concentration of free tubulin is high enough to grow a microtubule within the cell, then why would some of them be shrinking at the same time that others are growing? And that's where GTP comes in. And I'll try to explain that in the next few slides. Here's just another movie, again, showing ends of microtubules at the cell edge growing and shrinking constantly. So they're always doing this. And the dynamics by which they do this depend on a number of factors. They depend on the state of the cell cycle, and they depend on a whole bunch of different proteins that interact with microtubules. This constant growing and shrinking of microtubules is called dynamic instability. Any given microtubule can be in the process of growing or shrinking. These dynamics are driven by hydrolysis of the GTP that's bound to the alpha tubulin. This shows an example of the length of a given microtubule over time. So You can imagine that if you took a movie like the ones I just showed you, which maybe you didn't see, and you actually measured the length of an individual microtubule, it might get longer and then suddenly start getting shorter and then longer and shorter and so forth. What researchers realized is that they could characterize the growth rate of microtubules and the disassembly rate, and also the frequency with which an individual microtubule switches from growing to shrinking, which is called catastrophe, and the frequency with which it changes from shrinking to growing, which is called rescue. And it turns out that there are different factors within the cell that control these different parameters, how fast they grow, how frequently they switch from growing to shrinking, and so forth. This shows how this transition between growing and shrinking actually happens. Here are the alpha beta subunits of the growing microtubule.
The beta subunit is bound to GTP.
And that enables these subunits to add on to the plus end of a growing microtubule. And they form the structure that's called a GTP cap, because the end of the microtubule is highly enriched for
GTP-bound beta tubulin. But over time, this GTPase activity of beta tubulin hydrolyzes GTP.
And that destabilizes the microtubule. The hydrolysis of GTP to GDP causes the microtubule dimer to kink a little bit. And so it makes this kind of want to fray and fall apart. If the GTP cap, which stabilizes the end of the microtubule, is lost, if enough of the GTP is hydrolyzed as it's growing, if the rate of hydrolysis exceeds the rate of growth, then you don't have a stable cap anymore, and the protofilaments at the cap start to bend and start to disassemble. And that's how the microtubule starts to come apart, and then it releases these free subunits from the plus end and shrinks rapidly. The rate at which this hydrolysis happens and thus depolymerization of the microtubule happens, as I said, is controlled by other proteins that interact with microtubules that are collectively called MAPs, microtubule-associated proteins. This just shows that in a non-dividing cell, these radial arrays of microtubules actually play an extremely important role in trafficking of proteins, which I talked about last week. So we talked about vesicle trafficking, how they're constantly vesicles budding off of the endoplasmic reticulum, going to the Golgi, and then going from the Golgi to the cell membrane and various parts of the cell. And I mentioned at that time that these vesicles are trafficking by walking along individual microtubules. And so as vesicles move and bud with the Golgi, they move toward the minus end of microtubules. And so the Golgi winds up concentrating right next to the microtubule organizing center. And conversely, as vesicles traffic outward toward the cell membrane, they walk toward the plus end of microtubules. And they do that by attaching to motors that have ability to walk toward the minus end or the plus end, depending on what kind of motor they are. This just shows an image of this where the Golgi apparatus is co-localized with the sort of nexus of microtubules in the cell, and the endoplasmic reticulum gets dispersed throughout the cell by moving outward from the nuclear envelope toward the cell periphery, but it doesn't all concentrate there. It stays throughout the whole cell. This shows the specialized structure of a neuron, which can have axons that go up to sometimes 2 meters long. So for example, the neurons that extend to your big toes from your brain can be meters long. And they have a cell body where the nucleus is, but the axons can project great distances from the cell body. So a lot of the synthesis of cellular material happens in the cell body and has to be trafficked to the axon terminal, and that happens by movement of motors along microtubules. It would take decades for molecules to diffuse all the way to the end of the axon, but by walking along microtubules, they can move in a matter of hours to days over many meters in length. So this actually shows stills from a recording of vesicles in a nerve fiber moving along individual microtubules, and you can track them by a variety of different methods. I think I should skip the next slide because it's a movie and I'm worried it's going to crash, but it just shows that that microtubules, that their growth and shrinkage change dramatically between interphase and mitosis. And that is, as I mentioned, controlled by the behavior of microtubule-associated proteins that tune the growth and shrinkage rates of microtubules. And there are different classes. So I just talked about different ways that microtubule-associated proteins can stabilize microtubules by binding either to the side of the microtubules, those are called lattice binding, or by binding to the end by preventing GTP hydrolysis, or by physically stabilizing the end. And then conversely, there are factors that promote depolymerization from the end, or that actually cut microtubules in the middle and sever them, and that causes them to depolymerize since there's no GTP cap. This slide just mentions a few drugs that are used both in research and in medicine that affect the dynamics of microtubules. So these are natural products.
Taxol comes from the bark of a Pacific yew tree, and colchicine and colcimid and vinblastine and vincristin are derived from compounds that come from different flowering plants. I don't think it's clear why these plants make compounds that target microtubule dynamics. Taxol binds to beta-tubulin in a way that antagonizes its GTPase activity, so it actually stabilizes microtubules by inhibiting GTP hydrolysis. But these other drugs have sort of the opposite effect. They bind to dimers of alpha and beta-tubulin and inhibit their polymerization. So they favor the depolymerized state of tubulin and thereby destabilize microtubules. Taxol is well known as a chemotherapeutic drug. It's often given to cancer patients because at low doses it can inhibit the microtubule dynamics that are important for assembly and function of the mitotic spindle, which we haven't yet talked about, but we'll talk about on Wednesday. And by doing that, it preferentially inhibits dividing cells, which is often the way that chemotherapeutic agents work. Vinblastine and vincristin are also used to treat certain kinds of lymphomas, and I don't really know why, but it has something to do with their effect on lysosome stability.
When people take Taxol to inhibit the growth of cancer cells, they also suffer side effects, hair loss and intestinal problems that have to do with the failure of like the hair cells and the intestinal cells to regenerate those tissues. How do things move along microtubules? So they move through the action of microtubule motors. There are two main types of motors. Do stabilizing microtubules allow them to grow more instead of disassembling? Yes. In the case of Taxol, if you use it at a low dose, it will stabilize microtubules, but it won't actually impair their function in vesicle trafficking, but it does impair the ability of the cell to very rapidly remodel the microtubules, which is essential for cell division. Two major classes of microtubule motors. There's one major minus end-directed motor, which is called dynein. And dynein is an immensely complex motor that's made of a whole bunch of subunits that I'm not going to talk about in any detail, but you should be aware that it is a motor that walks along microtubules and preferentially goes toward the minus end. So it tends to walk, from the cell cortex toward the microtubule organizing center. There's a huge family of kinesins. Most of them are plus end-directed motors, but some are also minus end-directed motors. So as you might imagine, motors require energy. Kinesin gets that energy by hydrolyzing ATP. So it has these two globular ends. One binds ATP, and in the course of hydrolyzing ATP, it undergoes a power stroke, which means a conformational change that repositions it, and then it rebinds to the microtubule. And that enables it to walk in a hand-over-hand fashion along the microtubules. And I am not going to be able to play movies, unfortunately. What this shows is motors walking along microtubules, and in this case, they're tethered to vesicles that are carrying various lipids and proteins inside. In addition to mediating intracellular movement, microtubules can mediate the movement of cells by causing the beating of cilia and flagella. So cilia and flagella are actually very similar or even homologous structures, that there's not really a strict delineation between them. Cilia tend to be shorter and flagella are longer, and we talk about sperm flagella and the cilia of the inner ear and so forth. But these are filamentous structures that grow out from the cell surface, and they have microtubules inside them. And they enable things like sperm propulsion. For many organisms, they're involved in eating because they sweep liquid across the cell and enable cells to capture prey. And the way that these are organized internally enables the action of dynein, which is shown here in red, to cause beating. So that they have these arrays of microtubules In this case, it's what's called a 9+2 array. So there's 9 of these little doublet structures with 2 in the center. And they're tethered together by a linking protein. And because they're tethered together, the action of dining causes the microtubules to bend relative to each other rather than crawling. And that's what's illustrated here. It's just the movement of dining, that it would normally cause the microtubules to slide relative to each other, but because they're tethered together by this linking protein, they bend. I'm just going to briefly introduce actin in the last few minutes, but I won't go through everything that the chapter has to say about actin, which unfortunate because it is a really crucial component of cell biology. So I would encourage you to read about it, particularly the muscle contraction part. This is another version of the same slide. So this just highlights some of the key functions of actin within cells. As I mentioned at the beginning, that the filaments are thinner than microtubules, but they're much more flexible and elastic. And they can give rise to a number of different kinds of cell structures. These look like the cilia that I showed in some of the previous slides, but they're actually microvilli, which are projections of cells that are found in the intestines that form the high surface area needed for absorption of nutrients in the intestine. And they gain they have stability and rigidity due to the actin filaments inside them. Actin is also involved in a wide variety of different kinds of cell movement. So this shows a crawling cell with a structure called the lamellipodia at one end. And the green here is actin. Sorry, it's not red, but actin is not inherently red. It's just shown that way in the chapter. But by polymerizing at the leading edge of cells, actin drives membrane forward and enables cells to move. And actin forms all kinds of other protrusion structures at the edges of cells that enable them to engulf things and attach to substrates and so forth. So a lot of the stuff at the edge of cells is formed by actin. Actin also forms this ring structure in the center of a dividing cell that enables the cell to pinch off its membranes and undergo cytokinesis, and that requires the action of a motor called myosin. This shows something that I showed in the previous lecture, which is these yeast cells that are schmooing. They're putting out these projections that are involved in mating, and they do that by polymerizing actin at this edge of the cell to sort of grow in one direction. And of course, one of the most famous functions of actin together with myosin is the contraction of sarcomeres to contract muscles and enable large-scale movements. So actin is very versatile. It's present in different structures and different cell types. That is also, just like in the case of tubulin, controlled by a wide variety of actin binding proteins that interact with actin and affect how it polymerizes, where it polymerizes, and what else it interacts with. And actin can form 2 major kinds of networks. At the edge of cells, it tends to form what are called branching networks. They're branched because actin filaments grow out of the side of existing actin filaments by the binding of a nucleation factor to the side and then new growth. But they can also form much more linear structures like these stress fibers, and those are kind of catalyzed in a different way by nucleators that tend to nucleate long, straight actin filaments. So this is an actin filament. Unlike a microtubule, it's not hollow. It's a single filament formed by a single protein, which is just called actin. Sometimes it's called G-actin for globular actin when it's not in the filament. When it's in the filament, it's called F-actin or filamentous actin. And so this just shows how this protein, by associating with itself, forms a helical structure with 37 nanometers per full revolution of the helix. Just like microtubules, actin has polarity. So actin tends to bind to one end of a growing filament. The minus end, I mean, it's rarely free like this. The minus end, just like tubulin, is typically tethered to a nucleator. But in vitro, if you polymerize actin, it can form these filaments that are simultaneously growing at one end and losing subunits at the other end. Actin is an ATPase rather than a GTPase, but the principle of the cell using energy to control the growth and shrinkage of these filaments is very similar between microtubules and actin. And then I think this is the last slide that just shows some of the different organization of actin filaments and the different kinds of proteins that can interact with actin to control its organization and behavior. I sort of mentioned in passing that there are nucleating proteins. So just like Microtubules have gamma Turk. Actin has two different kinds of nucleators, formin, which tends to trigger the polymerization of straight filaments, and ARP23, which binds to the side of an actin filament, and so it nucleates branched networks. The main thing I said is that I'm not going to go into detail about the different kinds of actin binding proteins and how they regulate actin behavior, but I would suggest that you read about them because it's the kind of thing that could easily show up on the MCAT or other tests of general knowledge. The material in today's lecture is not going to be on the big quiz this week. For the final, you should know microtubules and microtubule associated activities, and I will not focus on actin. Is the final cumulative? Yes, it is. Roughly 2/3 of it is on my section of the course, and then we still have a couple of lectures from the other two faculty instructors coming up. So can you explain what she was saying for mother and daughter centrioles? I just said that centrioles have a replication cycle, and each cell has a pair of centrioles. And what happens during S phase is that each of those centrioles grows a daughter centriole from it. And so then you have two pairs of centrioles, and those pairs split to form the spindle poles in mitosis. So I'll have a little bit more to say about that on Wednesday. The older centriole is the mother and the younger one's the daughter. Do microtubules grow and disassemble only from the plus end, and actin grow just from the plus end and disassemble from the minus end? I showed you a figure from the textbook, which is often shown, in which actin is doing what's called treadmilling, which means you have an actin filament that's simultaneously growing from the plus end and shrinking from the minus end. And so it stays like steady state, stays the same length. But in practice, in cells, that's not really what happens. Just like microtubules, actin grows from nucleating proteins. And so the nucleating proteins are at the minus end of the actin filament, and the plus ends are growing. So I don't know why textbooks always show that, but they do. The last question was about the final, and so hopefully you saw the message from Nicole that there was an alarming prevalence of cheating on the midterm that she was able to identify and document, and that's part of why we are reserving judgment about how to offer the final, because if we offer it asynchronously, it increases the potential for people to share answers and so forth. So my preference would be to give it synchronously, but I don't want to penalize those of you who are in different time zones. So we will still figure that out. We can use a lockdown browser and a webcam. The campus has announced that they are trying to get a lockdown browser, which simply, prevents you from opening other tabs or using other applications while taking the final. I think the use of webcam proctoring is not supported by the campus because it's an invasion of privacy, I think appropriately so. And so the problem is that even a lockdown browser is not a guarantee that people won't discuss things, that they've seen with other students who haven't taken the exam and so forth. So it would probably be easier if we can give it all at the same time. Does that mean it won't be open note? It will be open note. You are allowed to refer to your notes and so forth, but as with the last midterm, you should not count on being able to look up the answer as you take the test, because the test will not, you won't have enough time to do the test if you have to look everything up. Yeah, so it will be open book and open note, but we're going to hopefully design it in a way that you can't just Google the answers. And if we do have a lockdown browser, it would make it harder to do that, but it wouldn't totally prevent that because you can always use another device. We want the exam to be as meaningful an assessment of what you've actually learned as possible. That means that we may have to prevent people from sharing information about what's on the exam. I will have office hours tomorrow. I'm happy to go through anything. I will try to either re-record today's lecture or at least patch up the parts that failed again. Will it still be okay to use online notes like Google Docs? Yeah, you can keep your notes however you like. What you are not allowed to do is share any information about the exam, about the questions that you've seen with your classmates. And you really should be relying on your own notes and your own knowledge rather than other people's to take the exam. I did switch tabs during last exam to use my online notes. Will that be okay? If the campus implements a lockdown browser, you will not be able to do that on one device. It will restrict you to one window and one application. So you might want to have an alternate way. You might want to print out your notes or something like that. I don't know if that's going to happen before the final, though. Can you just reduce the time to a day or 8 hours? We will discuss it. Yeah. As I said, it really depends on the resolution of the issues with the last midterm and also how the big quiz goes this week. I mean, we really want to be as fair as possible to the people who are actually studying and doing the work required to learn the material. Is the big quiz on B courses? It will be, yes. And we announced that it would be asynchronous. It will be, we plan to give it from Thursday noon to Friday evening. But again, if there are instances where we can detect people have worked together or shared information, that will not be good and it will not be tolerated. Another one of my classes had 2 offerings for the time zone problem. There were different versions, but each one had their own time slot. Yeah, I mean, that's another way we may resolve it, but that requires us to write 2 complete exams that are equal in difficulty, and that is a lot of work. So that may or may not be the best solution.